Assalamualaikum warahmatullahi wabarakatuh. I am Madam Nazira Zubair. In this video, we're going to focus on the adaptive immune response. For the adaptive immune response, there are two types. One is cell-mediated immune response and another one is humoral-mediated immune response. But I'm going to focus on the cell-mediated immune response. For the cell-mediated immune response, there are two types of cells that are very important, which are the T helper cells and T cytotoxic T cells. But for T helper cells, it's very important as well to trigger both the humoral and cell-mediated immune response.
The learning outcome, or LO, for this video is students should be able to describe the mechanism of the cell-mediated immune response. This figure, which is taken from Balaji Campbell textbook, describes cell-mediated immunity and humoral immunity. In humoral immune response, antibodies help to eliminate antigens by promoting phagocytosis and complement-mediated lysis. In the cell-mediated immune response, specialized T cells destroy infected host cells. Helper T cell triggers both the humoral and cell-mediated immune response. There are two requirements must be met for helper T cell to activate adaptive immune response. Firstly, a foreign molecule must be present that can bind specifically to the antigen receptor of the T cell. Secondly, this antigen must be displayed on the surface of an APC or antigen presenting cell. APC can be a dendritic, macrophage, or B cell. In this figure, APC is a dendritic cell. Helper T cells or TH cells interact with the antigen fragments displayed by class II MHC molecules on the surface of antigen-presenting cell. Activated TH cells secrete chemicals or cytokines that stimulate other lymphocytes. In the cell-mediated immune response, activated cytotoxic T cells trigger destruction of infected cells.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen.
Alright, next we're going to describe and discuss the cell-mediated response using a figure taken from Solomon Biology textbook. TH cells are activated by the foreign antigen MHC class II antigen complex displayed on the surface of APCs. Once activated, TH cell increases in size and divides by mitosis. Then the clone of competent TH cells are produced. Some TH cells attract macrophages to the site of infection and promote the destruction of intracellular pathogens. Others function mainly in antibody-mediated immunity. Right, next we'll take a look at TC. Once activated, a TC cell increases in size and gives rise to a clone. Activated TC cells are the effector cells that make up the cell infantry. They leave the lymph nodes and make their way to the infected area where they destroy target cells within seconds after contact. When a TC cell combines with antigen on the surface of the target cell, it destroys it in much the same way that NK cells destroy their target cells. The TC secretes perforins and granzymes that perforate the plasma membrane of the target cell and induce it to kill itself by apoptosis. After releasing cytotoxic substances, the T cell disengages itself from its victim cell and seeks out a new target. This sequence is summarized as follows. Virus invades body cell, then infected cell displays foreign antigen MHC class I antigen complex, then specific TC cell activated by this complex, then going to form clone of TC cells. Then after clone of TC cells produce, TC cells migrate to area of infection. And lastly, the TC cells release proteins that stimulate destruction of target cells. Next, we're going to discuss on the figure taken from Campbell biology textbook. This figure is on the killing action of cytotoxic T cells or TC cells on an infected host cell. Firstly, an activated cytotoxic T cells binds to a class I MHC antigen fragment complex on an infected cell via its antigen receptor and an accessory protein called CD8. Secondly, the T cell releases perforin molecules which form pores in the infected cell membrane, and granzymes, the enzymes that break down proteins. Granzymes enter the infected cell by endocytosis. Lastly, the granzymes initiate apoptosis within the infected cell, leading to fragmentation of the nucleus and cytoplasm and eventual cell death. The released cytotoxic T cell can attack other infected cells.
When a virus infects a cell and synthesizes viral proteins, some of these proteins are degraded to peptide fragments. These peptide fragments are complexed with class I MHCs and displayed on the surface of the infected cell. Cytotoxic T cells interact with the virus-infected cells by recognizing both the viral antigen and the class I MHC. The cytotoxic T cell then releases cytotoxins which induce apoptosis in the infected cell and perforin which causes perforations in the cell membrane. Self-proteins are also degraded and the fragments presented on the surface via class I MHCs. This is not shown. Fragments of self-proteins are not recognized by cytotoxic T cells. Cytotoxic T cells remain intact, detach from the infected cell, and move on to other target cells.
This last figure, taken from Campbell biology textbook, summarizes adaptive immunity, reviews events that initiate humoral and cell-mediated immune responses, and highlights the central role of the helper T cell in both humoral and cell-mediated responses. Both the humoral and cell-mediated responses can include primary and secondary immune responses. Memory cells of each type, helper T cell, B cell, and cytotoxic T cell, enable the secondary response. For example, when body fluids are reinfected by a pathogen encountered previously, memory B cells and memory helper T cells initiate a secondary humoral response.
Alhamdulillah, in this video, we're able to discuss, describe, and illustrate cell-mediated immune response, and hopefully later you're able to distinguish between cell-mediated immune response and humoral-mediated immune response of the adaptive immune system. Until we meet again, Assalamualaikum warahmatullahi wabarakatuh.